friend of Professor Dr. Muhammad Azzam Kayasah from United Arab Emirates. Dr. Kayasah is a consultant for gastroenterology, and he is a founder of Kayasah Gastroenterology Clinic and the founder of president of Virtual Microbiome Innovation Center and clinical research for UAE Microbiome Project, and also the virtual Dubai Endoscopy Forum and the clinical research Holistic Microbiome Signature Project. Dr. Kayasah, we are waiting for this lecture since long time, and you have questions at the end from many audience, so please start.
Thank you very much. Bismillah ar-Rahman ar-Rahim wa salatu wa salamu ala Sayyidina Muhammad alladhi bu'isa rahmatan lil alamin. It's my pleasure to be with you today because, you know, in Arabic we say ختامها مسك. إن شاء الله حتكون مسك الختام. طبعا today I'm talking about our microbiome signature, and you know what happened now in this session, what I call it IBD state of art session. All my colleagues cover all the risk stratification model, clinical scores by Dr. Tawil, biomarker score, endoscopy score by Susanna, and in addition to some also the complication by the colorectal cancer complication by Dr. Yusuf Ajlouni. Now my talk only about the microbiome score and the IBD microbiome signature and how we can treat also IBD by microbiome-based therapy. This is my talk on microbiome signature mapping. First, the concept of our microbiome signature. Second, I'll pass by IBD-Risk stratification model. Third, by some slide about UAE IBD microbiome signature project, and the last take-home message, and it will be surprise. You see the history. This is not recently they are talking about the microbiome. If you see here, since 1894, they are talking about periodontitis and what is going on, what is the relation between oral disease and rheumatoid arthritis. And after that, on 1991, the H. pylori and the history of H. pylori and how it is and what is the most important. In 2001, you see, come something called by Joshua microbiome. And this is the first time it's mentioned microbiome. And after that, on HMB, Human Microbiome Project, on 2007 and 2008, and Fecal Microbiota Transplantation, it come on 2015. And nowadays, starting from this year till 10 years coming, you'll see what is the microbiome pathogenesis in IBD, what is the precious medicine in IBD based on microbiome, and how we can treat IBD based by targeted microbiome modification. And you see, can we prevent IBD? In my opinion, I believe of prevention of IBD. When we can, if we work together on microbiome signature, we can prevent IBD as early as possible before it reaches to the symptoms. You see, I start HMB, Human Microbiome Project, start on 2008. Just 10 years later, I start to talk about UAE Gut Project and Syrian Gut Project. I hope this year I succeed to start about UAE Gut Project. Why we want to start today before tomorrow? Because as you see, American Gut Project and British Gut Project try to merge together to make something called the Microceta Initiative. Why? Because they want to collect all the samples from all over the world to study the microbiome and to be pioneer in this field. Why not to start to be our own pioneer in our Middle East and in Pan-Arab, in Africa, in our area? Holistic microbiome, it can be human microbiome, it can be plant microbiome, it can be animal microbiome, it can be soil microbiome, and it can be water microbiome. And you know, I put this one, Earth Microbiome Project. There is Earth Microbiome Project. And you see, Earth is the same like your Earth, and your target is research. That's meaning we can do under your society, we can do a lot on microbiome project. I hope we can succeed to do something starting today. Everybody know we are microbiome. We have, if we have 20,000 human gene, we have 2 million to 20 million microbial genes. That means up to now we study only 1% of whole genes what we have, and we neglected 99%. And if you see, this is a mucosa of the intestine, and this is the microbiome space life. I call it microbiome space life. This is, we cannot see it by endoscopy, we cannot see it by capsule endoscopy, we can see it only by special technique. I'll mention it later. Microbiome, it can be, this is the microbiome signature. I will go in details later. It can be gut microbiome, and it can be oral microbiome, it can be placenta microbiome, vagina microbiome, skin microbiome, chest microbiome, and each one is different from each other. And all this, it can cause, all this disease can be due dysbiosis, due to altered microbiome balance. Some vocabulary about the microbiome. You know, microbiota is the organisms. Metagenome is the genes of the microbiota. Metatranscriptome is the RNA. And metaprotome is the proteins. And metabolome, metabonome is the metabolite. And metataxon is 16S RNA gene, which is the most important. We are studying it a lot nowadays. But we should differentiate between bacterial community, which is, we call it microbiome, and viral community, which is called it virobiome, and fungal community, which is called microbiome. But we are talking, what you call it, as later, we can say microbiome, it can include all, but we should differentiate between all this type. Everybody from us, you, Dr. Burai, Professor Rida, all the balanists, we have in our microbiome signature system factory, starting from mouse up to the anus. That means daily we are product a lot of things, which is, if we really can do proper, can we reshape our microbiome signature, we can do a lot for our community. Any disease, it is 4D parameter in health and disease, and this is there is 3 parameters in addition to the host. There is gene, البصمة الجينية, there is البصمة البيئية, والميكروبيوم البصمة الميكروبومية. All this together, it will interplay together to make either health or disease. And you see, if there is obiosis, this is what I mean by obiosis. I will not go in details. And what is and this is mean health. If there is dysbiosis, this is what is the balance of the harmful opportunity is more than beneficial communals, the good microbiota, and this is, you call it gut microbial dysbiosis, and all this, it can be due to gut microbiota dysbiosis. We have in our mouse, as I mentioned, we have microbiome signature, which is, this is the bacteria, the picture of microbiome signature in health. And this is the most important nowadays, especially we are talking about IBD. IBD starts from the mouse. If really we have proper oral microbiome signature, we will protect our self and our patient and our children from IBD, because we have our microbiome is our immunity system. That's meaning, as you see here, I won't go in details in this because it will take a long time. That's meaning any disturbance in oral on our microbiome signature, it will make disturbance in the mucosa and in the intestinal mucosa or the mucosa or oral mucosa, and it will start to have the immunity disturbance, and it will start to have the disease. Can we protect the IBD? Yes. Can we treat IBD? Yes. How? If we interfere as early as possible, especially if it is preclinical. If we know what is the IBD microbiome signature and how we can make it in a proper way, and that's meaning if we can reshape our IBD microbiome signature and to be healthy, that's meaning we can prevent the IBD. All my colleagues today are talking about IBD-Risk stratification model. As you see, clinical scores, they already mentioned it. Biomarker, they already mentioned it. Advanced novel bioimaging score and therapeutic response rate score. Why? To differentiate between low-risk group and high-risk group. And the biomarker, this is my talk. I will go in details in this. IBD-Risk stratification model, microbiomarker scores, what I call it IBD microbiome signature. It can be differentiated between this is healthy control, and this is ulcerative colitis, and this is Crohn's disease. And you see the difference. That means we can differentiate by the level of microbiome between ulcerative colitis and Crohn's disease, and which I call it ulcerative colitis microbiome signature and CD or Crohn's disease microbiome signature. And in each one, ulcerative disease microbiome signature, this is a picture of the microbiome mapping, something up, something down. That's meaning we have something which is, we can test it by invasive test, and we can reach to a diagnostic tool and also a therapeutic tool if we based on microbiome-based precious medicine in IBD. What is the benefit of IBD microbiome signature? It can be for prediction, for diagnosis, for prevention, and for treatment. And there is a lot of study nowadays in America about 3 study gene or 3 project since long time. They are working on this gene project, Meconium study, and the other one, the DSR. This is in the military, and this is the one in by Charlie Lee predict study. Also, they are studying what is the role of the microbiome. This is in United Kingdom. That's meaning what is the benefit of IBD microbiome signature? It can be a screening tool. A screening tool, yes, we can reach a screening tool by IBD microbiome signature. This is study done in pediatric, and you see, if you saw, this is a control. This is a healthy control, and this is healthy control. There is no IBD in the mother, and this is the baby of IBD mother. If you saw the conclusion, babies born to mother with IBD have less beneficial microbiome, which can adversely affect the development of their immune system. That's meaning if we reach to this signature, we can really change the world of IBD in our area. Because IBD in Middle East is different from IBD in the United States, different from IBD in Europe, different from IBD in Asia. That's meaning we should study our IBD microbiome signature, especially in our children. When I start a couple of years before about the concept of the project, FUE and IBD microbiome signature, of course, there is a lot of things we can do. We start also to do some study about antimicrobial resistant microbiome signature, IBS, NAFLD, and now there is muffled, we call it MAFLD, diabetes mellitus, obesity, autism, colorectal cancer, as mentioned by Dr. Yusuf Jaluni, breast cancer, pancreatic cancer, and the others. How we can test the microbiome. That's meaning how we can analyze the microbiome. This is the most important slides. If you see, we can test our microbiome either in stool or urine or blood or tissue, either to test 16S RNA gene or metagenomics or metatranscriptomics or metabolomics. That meaning this is more cheaper than the other. This is other is more expensive. But nowadays we are starting to do something about 16S RNA gene, and especially just last week we started our project in COVID-19 patient and how we can really reach to prevent or to treat COVID-19 patient before we have the vaccine. This is I mentioned by Professor Charlie Lee from United Kingdom. This is how we can start as early as possible to study our patients and to study in the family of our patient and to find the IBD microbiome signature and to protect our patient from having proper disease. And this is another slides about there is factor which is implicated in IBD. There is a lot of factor, diet, stress, social status, and a lot. And how it can be interfere with microbiome dysbiosis, and it can be in the IBD and flare up of IBD and complication of IBD. There is another test in metabolite. We call it short chain fatty acid. And this is, there is 3 parameter, acetate, butyrate, and propionate, and this can be tested also in the blood. And even there is nowadays, there is special capsule will come in the near future. It can be tested all this by in the stool. You see, this is the pathogenesis. I will not go in detail in pathogenesis, how this metabolite, it can make a lot of damage on immunity, and it can make a lot of initiated the disease in IBD patients. The last part of my talk about the IBD-Risk stratification model, therapeutic response rate score. And you see in therapeutic response rate score, as my colleague mentioned about the immune modification or immune suppression, and sometimes about the surgery. But my role here to insist how I can do microbiota manipulation to change the picture of IBD, either by dietitian, by dietary nutrition treatment, or antibiotic, or postbiotic, prebiotic, probiotic, intestinal microbiota transfer, or bacterial-derived protein. And this is just slides to make the take-home message from this part. And the most important, you see, a good diet is the best prescription. If really we know how to eat, if we really teach our patient how to eat, he will be fantastic. The MELODY trial also, this is, you call it modulating early life microbiome through dietary intervention in Crohn's disease. And this is really very important. A lot of, what you call it, a lot of results have it from this, how we can manipulate the Crohn's disease patient if really describe it or discover it as early as possible. Just some slide about fecal microbiota transplantation. Nowadays, to be more soft, no more calling fecal microbiota transplantation. We call it intestinal microbiota transplantation. And there is nowadays stool bank, and nowadays there is putty, and there is a lot of things which is really, we should take care of this to know exactly how we can benefit for ourselves and our patient. Tomorrow, this picture is from Dubai, from our lovely Dubai. You know, tomorrow World Microbiome Day, it will be 27th June. And this Microbiome Day, I mean holistic microbiome for human, plant, animal, and all. I hope we can work together to celebrate. And today I am celebrating with you our World Microbiome Day, and it will be mentioned in their website. Really, you are the intelligent player today. If we can do some project like this, if we announce today our project, Pan Arab IBD Microbiome Signature project will be really breakthrough to do such such things for our future and for our healthy society. We should not forget about vitamin D microbiome signature, which is very important in IBD. This is another project we can work on it. And this is my last slide about take-home message. Really, Crohn's disease, ulcerative colitis share many epidemiological, immunological, therapeutical, and clinical features, but they are two distinct subtypes of IBD at the microbiome level. If we propose the microbiome markers to discriminate between these two types, really we can, by IBD microbiome signature, really we can have breakthrough for our future. Thank you very much for your listening.
Thank you, Professor Azzam. It's really interesting lectures. It grasps interest from audience everywhere in the world. Allow us to share the slides if possible. And
yeah, of course, I'll do it.
Thank you very much. And I would like to ask our expert panelist, Professor Ashraf Brady.
Thank you.
Professor.
Thank you, Professor. First, I'd like to thank the Society and the Conference Board and the Organizing Committee for inviting me for this valuable scientific meeting. And thank you, Professor Azzam, for this elegant and informative talk. I have two questions. The first one, you mentioned that we could prevent IBD. So do you believe in the hygiene hypothesis, which suggests that the lack of early childhood exposure to symbiotic microorganisms or helminthes parasite increases susceptibility to immune-mediated disease in later life? As we see that the percentage of IBD cases increase, especially in the industrially countries and developed countries. Second question, you know that you have mentioned that many factors which can shape the composition of gut microbiota, such as host genetic, diet, drugs, comorbid diseases, and lifestyle, all of these can shape the composition of gut microbiome. How can we get a signature for IBD patients in the presence of all of these heterogenic factors? Thank you.
Please unmute your mic, Dr. Azzam.
Unmute, we can hear you.
Yeah, it's okay now?
Yeah.
Yes.
Okay, how we can do it? First, it is not easy. We should start to do from now to collect the stool to know from healthy pediatric, healthy children, and to know the healthy children from IBD family patients, and to know the IBD microbiome signature. Because this is not, as you say, it's not easy, it's difficult, but it should be work all together to find what is the best microbiota mapping in IBD patients. I know, you know, the microbiome, as I mentioned in one slide, it can be bacterial, can be fungal, can be virus, and can be parasites. That's meaning we should know what is the beneficial for our children to have as early as possible their parasite infestation in their early life, because there is hashtag called keep dirty for children, but not now in COVID area. This is, it can be what you mentioned. The first question is parasites can be induce the immune system. It can be, but it needs to be studied.
Thank you. Thank you.
I need to hear from Professor Rida.
Yes, Assalamu alaikum. Welcome, welcome, Dr. Azzam. Until first time we meet at 2016 at Dubai. Not yet started. Yes, not yet started the practice of microbiome in any disease until now.
No. You mean as a project, so, as IBD, as microbiome signature?
Yeah, you didn't do any studies, short pilot study like that I did in HCV patient, at least to know the profile of normal people at your area. This is what we need.
No, you are right. You are right, Dr. Rida. Still, we didn't do it because up to now we didn't find any sponsor to do such things, and we should find a special lab. Nowadays, I'm working just recently last week with Khalifa University in Abu Dhabi, and I hope we can succeed in near future to have what you are telling, to have about IBD in healthy people, to have healthy microbiome signature, and to have IBD microbiome signature, to have muffled microbiome signature. I hope in the near future. Now I start the first track, and I hope I can succeed in near future with you. What is your opinion about Pan-Arab IBD microbiome project?
Yeah, you already added me with you. I don't know what the progress. This is with you, not me. You are the one who started. Yes.
I know, but now I'm waiting for Professor Youssef Ajlouni to support us in Pan-Arab.
Now I have a project now about microbiome in fatty liver. Later on, I will let you know the result. Inshallah.
Very good. All the best. All the best. All the best.
Inshallah. Inshallah. Thanks a lot, Dr. Muhammad.
Thank you. Thank you.
Thank you very much, Dr. Reda, Dr. Azzam. May I ask Dr. Dina Ismail if she's hearing me, please, to unmute your mic?
Yes.
Yeah, Dr. Dina. So we have discussed a lot of things in this session, starting from the extraintestinal manifestation from inflammatory bowel disease and the colon cancer prevention, and going to the monitoring, and after that to the microbiome. So what do you think if we have this high level of practice with these lectures, will we push the IBD practice in Egypt to a better place?
Yes, I think that after this beautiful session, we can push our young colleagues and us to perform new research work on this part. I mean, what Dr. Muhammad, Professor Muhammad introduced was very, very promising. And I think we should collaborate in different centers here in Egypt and in UAE and the different Arab countries to perform excellent multi-center research and have excellent results. And this, I think, will increase our knowledge to help the patients. Yes.
Thank you very much.
Dr. Burai, can we announce something today from your side? Because we need something earth, earth.
Yeah, please.
Now, because I want you from, I put the slides about Pan-Arab. Can you do something today, or we can announce something as Pan-Arab IBD microbiome signature under earth?
I support personally the idea, and I promise to put it in our next council meeting. And
thank you.
Yeah, we will ask Professor Ajlouni also if